Oh, that's weird. Why does it do that? There we go. Amino acids are coded by mRNA base sequences. Translation converts mRNA messages into polypeptides. Oh, it just, oh yeah, I can't do that. It always does this when I try to go back. Here we go. Translation converts mRNA message into a polypeptide or protein. Amino acids are coded by mRNA base sequences. So mRNA has the code that creates the protein. And the translation is what converts that code into a protein, also called polypeptide. I wonder where Hannah is. I know she was here today. Huh. No, we'll talk more about this later on. So don't worry about drawing that for right now. So a codon is a name for 3 nucleotides in a row. And 3 nucleotides in a row code for an amino acid. So you can see right here, these 3 nucleotides, AUG, that's a codon. And that codes for one amino acid, in this case methionine, is what is coded when you have AUG. If you have CUU as the codon, that codon calls for leucine, a different kind of amino acid. Oh, sorry. There we go. So each codon has a unique letter sequence, 3-letter sequence, which calls for a certain amino acid. There's only 20 amino acids. There's a lot more combinations of codons. And so a lot of codons will code for the same amino acid. Usually about 3 or so will code for the same amino acid. So there is a little bit of overlap. But this is how mRNA builds proteins, because amino acids are the building blocks of proteins. And so by each codon calling for one amino acid, it builds the protein one block at a time to create that unique protein. Genetic code, which we're seeing right here in this chart, tells you what codon matches up with what amino acid. So this gives you the letter sequence, the 3-letter sequence of each amino acid, all the combinations possible, and what amino acid you would see from those 3-letter sequences. So you can see up here, if you have UUU as the codon, that calls for an amino acid called phenylalanine. But UUC also codes for the same amino acid. So this helps when there's mutations, when you have just one base letter difference. So you might have a mutation where U becomes a C. Well, in this case, that would not harm you at all. You wouldn't even know you have that because you still get the same amino acid in both cases. And so this helps in that situation. When you do have mistakes, sometimes they're not harmful mistakes.
What is a stop sign?
Oh, that's a good question. So you always have to have a start and a stop codon. So methionine right here triggers a start. And then the stop tells it to stop making the protein. So the proteins will start with methionine, and then they will end with a stop code, which tells it to stop making the protein. It's finished. And at that point, the protein detaches, and then it's transformed and rearranged to create the finished product.
So our protein is amino acid.
That's right. Yeah. So to read this chart, you can see right here, the first base right here, second base, and then third base. So if you're looking to see, what is GAC? What does GAC code for? Guys, is something wrong right there? So let's say we want to find GAC. How do we do that? Well, G is the first letter. So we're going to go over here to look for that. So G, what's next? A, second letter up here. So we know that we need to go this row, this column, and then C was the last one. We go to this row right here. So GAC, aspartic acid, is what GAC codes for. So that's how you use this chart. Genetic code right here. And what's really fascinating about this is that this is good for any cell you're looking at, whether it's a plant, animal, human, they all use the same exact code, which is really interesting and beneficial to us because that is true. We can take human DNA, human genes, like a gene that makes insulin in the human body, put that gene into a bacteria. That bacteria can use that gene to make insulin. And so we now have in laboratories bacteria that make insulin for humans. Before we had to get insulin from cows or from pigs, and it wasn't the best way to get insulin, but that's all we could do. But nowadays we can get insulin from bacteria in a factory, in a laboratory. And so it should be a lot cheaper. Now there's different reasons why the price is high right now, but it's a lot cheaper to do it that way than to get it from animals. It's also safer and better for us because this is actually human insulin and not insulin from a pig or a cow, which is a little bit different. And so we can do that because the bacteria genetic code is the same as a human genetic code. So the bacteria reads the same code that a human does, and it produces the same proteins if it has the same code. So we'll go into some more detail later, but just the main idea is what you need to know about this genetic code, just that each of these three-letter sequences gives you a certain amino acid, and those make up proteins, which then do many different things in your body. There are 3 stop codes, like Lucy pointed out. You have these right here, which tell the body to stop making the protein. So that's the end of the protein when you get to that point. So it has to know when to start and when to end the protein. And so methionine tells it to start, and then one of these 3 codons tell it to stop. And then there's one start codon, as I mentioned, methionine, AUG. That's a start signal. You might have heard of some of these before, like tryptophan. You might have heard of that. Do you know where you find tryptophan? Has anybody heard about that before? Turkey is one place you find tryptophan, and people think that that's why you get sleepy after eating turkey. It's somewhat of a myth. I mean, tryptophan can make you sleepy, but the amount that you get from turkey is not enough to really do that. It's more of just eating a big meal at Thanksgiving that makes you feel sleepy. It's not really the tryptophan that makes you do that. But people blame it on tryptophan when they get sleepy after eating turkey. Which one would you know about? If you're an athlete and you're taking protein powder, you might know some of these ones. Maybe cytosine, you might have seen that one in the protein powder, or glutamine, or creatine. A change in the order in which codons are read changes the protein. So for the first one, it starts with C and it reads it as CGA, which gives you arginine, and then UAC, tyrosine, AGU, serine, and so on. But if you start with G right here, you get GAU, aspartic acid, and it's going to be totally different amino acids. So based on where you start, you're going to get very different codes, very different proteins. You don't need to draw these out. Just understand what this means, that wherever you start is important because your starting point dictates the rest, which will change the entire sequence if you start at a different place.
So how does your body know?
Oh, so the start codon methionine. So it's going to look for an AUG. An AUG tells it to start. Okay, so down here, this bottom bullet point, codons code for the same amino acid in nearly every organism. As I pointed out before, the genetic code is really the same for pretty much everything, whether it's a bacteria, plant, animal, or human. There's really no difference at all. There's a few minor differences in some species, but pretty much it's universal for every living organism. And that's why we can put, we can take out a gene from a jellyfish, put it to a cat, and the cat glows in the dark. Or the mouse you saw in the same thing. They took out the jellyfish gene and put it into the mouse to make it glow in the dark. We can transplant genes from different animals and we get the same result. The body reads it the same way in different animals or humans or plants. So we talked about codons, and then there's anticodons which attach to the codons. So amino acids have to be linked together to make the protein, and anticodons help to do that. So an anticodon is 3 nucleotides that attach to the codon. So anti can mean against on the opposite side. So anticodons are on the opposite side of the codons. That's what that means in this case. It means that it's on the opposite side of the codon in the strand. An anticodon holds 1 amino acid. So it complements mRNA. Complementary means that it's on the other side of. It goes with mRNA. An anticodon is carried by tRNA. So tRNA, called transfer RNA, moves to mRNA. The anticodon attaches to the codon. So if a codon were, say, CUG, the anticodon would just be the opposite. So if you have a C right here, you're going to have what letter over on this side? G. What about over here? So if this is a U, what would you have over here? A. And then what about this one? So what did I say before? I forget now. Let's just say over here we have an A. If we have an A on this side, what would you have on this side? A T. So if it were DNA, it'd be a T. Now this is RNA though, so instead of a T, what do you have? A U, exactly. Yeah.
So I thought the codon was the amino acid, or is it? Codon?
No, so the codon calls for an amino acid, but the codon itself is not the amino acid. A codon will tell the body to get an amino acid, but it's not itself the amino acid. So a codon is going to call for a tRNA, which holds the amino acid. So AUG will call for the codon, which is the reverse of that. So A would code for U, U would code for A, G would code for C. So you'd have on this side the opposite of those. And you'd have an anticodon, which would be U, A, C. And that would code for methionine right here.
So the tRNA is not made up of DNA, it's made up of RNA.
Well, so the tRNA is made of just RNA, right?
But is it like is it template the mRNA, not the DNA?
That's exactly right. Yes. After that, we're now in the ribosome. So tRNA, mRNA join together in the ribosome. Remember, the ribosome is a factory which makes the protein. So at this point, they join together to create the protein in the ribosome. The ribosome has 2 parts to it. It has 2 subunits. The smaller part is where mRNA is. Yeah, it'd be good to just like sketch, just a really rough sketch of what we're seeing here. You don't need to draw it perfectly, but just get the idea of what's happening here would be good. So the smaller part is just the strand of mRNA that goes along here like a ribbon. It just read through. And then the larger part up here, that's for tRNA. And you have 3 tRNAs which can come in here at any one time. And as they come in, they bring an amino acid in, which is then bound together to create the protein. So the large subunit holds the tRNA, which holds the amino acid. The small subunit holds the mRNA. And the ribosome itself is made out of RNA too. It's made out of rRNA. So you have the ribosome made of rRNA. You have tRNA that brings an amino acid. You have mRNA down here, which is the message, which tells it how to code for the protein. Everybody have this one down? And so ribosome just reads one gene at a time. So mRNA strand is just a strand of one gene from the DNA. And that one gene codes for a certain protein. And that's how genes are expressed, is by having one protein made at a time from that certain gene. This gives you a bigger picture of what's going on here. You have the ribosome right here in green. You have tRNA coming in to link with the mRNA. And it just reads down the strand. And as it reads down the strand, it's going this way. It's making a protein chain. This is the protein chain that's creating. So these little balls right here are amino acids, which are being brought in, and they're being joined to this strand. And as it goes down the mRNA strand, it gets bigger and bigger for the protein until it's detached and the protein is finished being made. And this mRNA came from the nucleus. Remember we talked about transcription before, where DNA is transcribed into RNA. And that's now out of the nucleus into the ribosome. That RNA is now read to create the protein that it calls for. So the complementary tRNA molecule binds to the exposed codon, bringing its amino acid close to the first amino acid. So it comes in over on this side, so there is on the right-hand side is an opening where the next tRNA comes in. And then this one at the left detaches. This one right here in the middle attaches this amino acid to the growing protein strand. When it moves down here, this one now comes over here, it detaches, this one comes to here, a new one comes in here. So it keeps on going from right to left. It keeps on moving this way. Yeah.
So that protein chain transferred over the next one.
Exactly. Yeah, it just keeps on growing the chain of amino acids as it moves forward. Yeah.
And when is the tRNA made? Is it made?
Oh, tRNA is just floating out in the cytoplasm. That comes from the food that you eat. And so as you eat, protein, your body breaks down to amino acids, which the cell then uses to make the tRNA. So everybody have these down? Okay. So there's not 3 ribosomes on here. It's just one ribosome, but just showing you what happens as it moves down, how you can see the chain keeps on growing as it moves down. So you just have one ribosome that constantly is moving down this mRNA strand to grow the protein chain. And this can be hundreds to thousands of base units long. It depends on the kind of gene you're talking about, but genes are mostly in the hundreds to low thousands as far as how many nucleotides it's going to go through to make a protein. Okay, so the ribosome, let me go back here, sorry. Yeah, we did that. Here we go. So the ribosome helps form a polypeptide bond between the amino acids. So polypeptide, that's just the name for a protein bond. It's a bond between amino acids, which creates the protein. So right here, you have a bond between amino acids, methionine and leucine. That is the peptide bond that joins them together to create the protein. And you can see right here how that happens. You have the start codon right here. The mRNA strand has AUG. tRNA has the anticodon, which is just the complementary pattern, UAC. And that codes for methionine. And that's the start amino acid. So that is the first amino acid you're going to always have for protein. Now, after it's finished, it can cut that out if it needs to. If it doesn't need a methionine at the end, it can just remove it to make the finished protein. But it always starts with the methionine. And as it moves down, you can see it adds the next one in here. So methionine is out here now. Leucine is the next one inward. And it'll keep on adding to the chain on this side. So this is the first one, second one, third one would be next here, and so on as it goes down. So the ribosome pulls the mRNA strand, one codon every time. So it jumps forward 3 nucleotide bases every time. So it goes from being right here to being right here, then to being right here. You each have a 3 nucleotide space for the next tRNA to come in. Can I add these ones down? Okay, so now it's empty on this side. The tRNA on this side gave a methionine to the amino acid chain. And once it's empty, the ribosome moves forward, it detaches from the mRNA strand, and it will get a new amino acid in the cell. But then a new one comes in here, a new tRNA comes in, and it calls the next one. In this case, cytosine is called next. Sorry, cysteine, not cytosine. A complementary tRNA molecule binds to the next exposed codon. And so it just keeps on adding in tRNA molecules that way. And it builds the chain of amino acids. You can see it along here. Eventually, it's going to find the stop codon. There's 3 different possibilities for that. But once it finds a stop codon, then the ribosome detaches and it just disassembles. And now the protein is finished. But then once the protein is attached, it might need to be further rearranged. Your proteins have different kinds of shapes they can take. And so just making the chain would not be enough for the amino acid. You have to do more than just make the chain. You have to also have the right shape of the protein. When you cook eggs, you are changing the protein shape, which changes the form of the egg from liquid into solid. So by adding heat to the egg, you are changing the shape of the protein, which changes its nature and what it does. So the protein shape is very important. And if that changes, then you have a different form. That's why a fever can be so dangerous for you. 108-degree fever and above changes the shape of your protein in your brain, which destroys, it basically fries your brain, literally fries your brain, just like an egg. You know, your protein in your brain has changed with the heat, and it's never going to be the same again. And so the shape of the protein determines its function. So it's important after you have the chain that it's assembled correctly in the right shape, or it won't work.
So what happens is anticodons afterwards? These could be recycled.
Oh, they're recycled. They grab new amino acids and just go back into the same thing over and over again. They're kind of like machines, like cars. You know, they transfer material and they go back for more material and keep on going back and forth, back and forth.
If you get anticodons from your food, then.
Can you get too much?
Well, you said you don't get anticodons from your food. Then what happens if you keep those and you get more?
Oh, well, eventually, I mean, your body might, if they get old, your body might tear them down and recycle them if they need to. And anything that's excess, your body just converts to waste. So if your body can't use it, it's waste in your body and it doesn't stay in your body. So whatever is excess your body can't use either converts to fat and stays in your body as fat, which can happen with protein, or it's just waste. Oh, are we already done with that? Okay, I guess it is the last slide. Okay, so for homework today, it's at section 8.5. Let me see. That's questions 1 through 5 on page 233. Page 233, questions 1 through 5.